Hey there, fellow microbe enthusiasts. Ready for another deep dive?
Always.
Today we're tackling a familiar foe, one that I'm sure keeps every medical microbiologist on their toes. Staphylococcus aureus.
Ah, yes. S. aureus. A classic.
And to guide us through this, we're tapping into Principles and Practice of Clinical Bacteriology, latest edition, of course.
Can't go wrong with that.
Our mission: to equip you with the latest need-to-know info on identifying, diagnosing, and most importantly, managing those tricky S. aureus infections.
Sounds like a plan. Where should we begin?
Well, let's start with a little trip down memory lane.
Oh, you mean back to Rosenbach in the 1880s?
Yep. I've always been curious about how they figured out this bug was different from the other micrococci way back then.
Oh, well, those grape-like clusters under the microscope were definitely a big giveaway. But Rosenbach also noticed some key differences in its growth patterns and the pigment it produced. That distinctive golden color was a real standout.
Right, the golden staph.
Exactly. Though, as we know now, that pigmentation can be a bit misleading sometimes.
Ah, you're talking about those sneaky small quality variants, the SCVs.
Precisely.
I remember this one case, a patient with a prosthetic joint.
Oh, those can be tricky.
Took forever to figure out it was an SCV causing a persistent infection. It was growing so slowly, and there was no golden pigment. So what triggers these SCVs to emerge in the first place?
That's a question researchers are still unraveling, but we know they can pop up after exposure to certain antibiotics, especially aminoglycosides like gentamicin.
Interesting.
It's thought that this might be a survival tactic for S. aureus, a way to hang on in the body despite those antibiotic attacks.
So it's like S. aureus is playing hide and seek with our immune system and our treatments.
Exactly, and that makes them a real challenge clinically. They're often behind those chronic or recurrent infections and can be much tougher to treat than their regular counterparts. Talk about resilient.
Speaking of resilience, S. aureus sure has a knack for sticking around, literally. It's got this whole arsenal of adhesins that let it cling to host tissues, and it even surfaces like medical devices. I read about a case of prosthetic valve endocarditis.
Oh, those are tough cases.
Caused by S. aureus latching onto the valve surface using its fibronectin-binding proteins.
Those fibronectin-binding proteins, or FNBPs, are some of S. aureus's most important tools. They play a crucial role in various infections.
Endocarditis, osteomyelitis, even wound infections.
Exactly. And what's fascinating is how they bind to fibronectin, a protein found in our own tissues. It's like S. aureus has figured out how to anchor itself directly to us.
So it's not just about invading, it's about establishing a base camp.
Right.
And once it gets a foothold, it can cause some real damage.
Oh, absolutely.
Yeah.
Alongside those adhesins, S. aureus produces a whole range of toxins and enzymes that contribute to its, shall we say, unfriendly nature.
It's a real multitasker.
You've got the superantigens capable of setting off a massive immune response, leading to conditions like toxic shock syndrome. And then there are the hemolysins, those red blood cell destroyers, and the exfoliative toxins, which, well, they cause the skin to peel away in conditions like staphylococcal scalded skin syndrome.
Okay, those are some serious weapons.
Indeed.
And of course, we can't forget about Panton-Valentine leukocidin, or PVL. It's been getting a lot of attention lately, especially with the rise of community-acquired MRSA.
You're absolutely right. PVL is a potent toxin that specifically targets white blood cells. Think of it as our first line of defense against infection.
Right.
It's often found in CA-MRSA strains and has been linked to more severe skin infections, necrotizing pneumonia, even sepsis. It's a definite red flag, especially in those community settings.
It's amazing how S. aureus can cause such a diverse range of clinical presentations. It's like it's constantly adapting its attack strategy.
That's what makes it such a fascinating and challenging organism to study. It's always evolving, finding new ways to thrive and unfortunately cause disease.
And that's just the tip of the iceberg. We've covered a lot of ground already, but there's much more to explore.
Absolutely. There's a whole world of clinical features and lab techniques waiting for us. We'll dive into those in part 2 of our deep dive.
Exactly. So for all you microbe mavens out there, stay tuned. We'll be back with more insights on Staphylococcus aureus in just a bit.
We'll see you then.
All right, microbe detectives, back for more S. aureus sleuthing.
Always up for a good microbial mystery.
We've unmasked its clever disguises, explored its sticky arsenal of adhesins, and even witnessed its toxin-fueled attacks. Now, let's shift gears and dive into the clinical world. What happens when this stealthy foe actually launches a full-blown infection?
Well, prepare to be amazed by S. aureus's versatility. It can cause a truly impressive range of clinical presentations.
So we need to be ready for anything. What are some of the most common signs and symptoms we encounter with these infections?
One of the most frequent presentations is skin and soft tissue infections. Think about those classic boils, the impetigo, cellulitis, the usual suspects. They can range from mild and localized to pretty severe, depending on the strain and the patient's overall health, of course.
Yeah, it's not always straightforward, is it?
Yeah.
I've seen cases that started looking like a simple skin infection but turned out to be much more serious, especially with patients who have underlying conditions like diabetes.
Absolutely. And S. aureus isn't a one-trick pony. It can cause infections deeper in the body, too. Septic arthritis, osteomyelitis.
Ouch, those sound painful.
They certainly can be. And those are the cases where accurate diagnosis and prompt treatment are critical.
Especially with antibiotic resistance on the rise, we definitely need to tackle that beast later on.
Agreed.
But before we go there, I want to circle back to those toxin-mediated diseases. Those can be pretty scary, right?
Oh, for sure. Toxic shock syndrome, or TSS, is a prime example. A life-threatening condition that can develop rapidly. It's often associated with specific strains of S. aureus that pump out those superantigen toxins.
TSS is definitely a clinical scenario that sends chills down my spine. Early recognition is absolutely crucial. And then there's staphylococcal food poisoning.
Yeah.
Maybe not as life-threatening, but definitely not a picnic either.
Not at all. Food poisoning is a classic example of how those S. aureus toxins can cause havoc even without a full-blown infection. It's all about those preformed enterotoxins that the bacteria release into food. Once those toxins get into the system, wow.
Nausea, vomiting, diarrhea, the whole 9 yards.
Exactly. Not a fun experience.
Speaking of not fun experiences, we have to talk about hospital-acquired infections. Those are a constant battle in healthcare settings, aren't they?
Absolutely. Nosocomial infections are a major concern, and S. aureus is one of the main culprits, especially for patients who are already vulnerable due to weakened immune systems or those with indwelling medical devices.
It's like S. aureus is an opportunist, always waiting for a chance to strike.
That's a perfect way to put it. And the types of infections we see in hospitals can be quite serious. Surgical site infections, pneumonia, bloodstream infections, endocarditis.
Sobering reminder of why infection control is so crucial.
Couldn't agree more.
But let's switch gears now and talk about how we actually identify and diagnose these S. aureus infections in the lab. Where do we begin?
Well, thankfully, we have a whole toolbox of reliable lab techniques at our disposal.
Good to hear.
It all starts with those classic methods, microscopy and culture.
Back to basics. I still remember the first time I saw those gram-positive koshis in clusters under the microscope. A real classic for S. aureus.
It is. And then we move on to culture to isolate the organism and get a definitive ID. Blood agar is our workhorse medium, and we can use selective media like mannitol salt agar to differentiate S. aureus from other Staphylococci.
Right. And then we've got our arsenal of biochemical tests to confirm the identification.
Precisely. The coagulase test is a classic most S. aureus strains produce coagulase, an enzyme that makes plasma clot. It's a quick and easy way to tell it apart from other Staphylococci that don't produce this enzyme.
Clever little buggers, leaving their telltale clots.
Indeed. And of course, there's the catalase test, DNase test, and even latex agglutination to detect specific S. aureus antigens.
We've got a whole detective kit.
Exactly. Accurate identification is crucial, especially when it comes to making those treatment decisions.
Speaking of treatment, we can't ignore that elephant in the room, can we? Antibiotic resistance.
You're right. It's the challenge that keeps on challenging. S. aureus is a master adapter, becoming resistant to many of our frontline antibiotics. It's a constant arms race.
Feels like we're always playing catch-up.
Sometimes it does.
So let's rewind a bit and talk about the big players in this resistance saga. I remember learning about penicillin resistance way back when. A classic example of how bacteria can outsmart our drugs.
It was. Penicillin resistance was one of the first hurdles we had to overcome, and it's primarily due to those beta-lactamases, enzymes that break down penicillin and other beta-lactam antibiotics.
So S. aureus built itself a shield against our penicillin arrows.
Quite effectively.
But we countered with methicillin, right?
We did, and for a while it was a game changer. But then, of course,
S. aureus outsmarted us again.
it did. And that's when methicillin-resistant S. aureus, or MRSA, entered the scene.
MRSA. 2 letters that strike fear into the hearts of many a healthcare professional. And it's not just limited to hospitals anymore, is it? We're seeing more and more community-acquired MRSA, or CAMRSA.
That's right. It's become a major public health concern.
So how does S. aureus pull off this methicillin resistance? Methicillin has a beta-lactam too, right? But I'm guessing the mechanism is different from those beta-lactams as we talked about earlier.
You're right. MRSA has a different trick up its sleeve. It all comes down to a gene called MECA, which codes for a modified penicillin-binding protein, or PBP.
Wait, PBPs. Those are the proteins that penicillin and other beta-lactams normally target, right? The ones essential for bacterial cell wall synthesis.
Exactly. But the altered PBP produced by MRSA has a low affinity for methicillin and other beta-lactams. They just can't bind effectively, rendering them
Useless.
essentially.
So S. aureus changed the locks on its doors, and our methicillin keys no longer fit. Clever.
Very clever. And to make matters even more complicated, the MECA gene is often carried on mobile genetic elements, meaning it can easily transfer between different S. aureus strains.
So it's not just individual strains developing resistance, it's this ability to share those resistance genes with each other.
Exactly. A reminder of how rapidly antibiotic resistance can spread, and how important it is to understand the epidemiology of S. aureus.
Epidemiology is key. But before we get into that, maybe it's time to shift gears again.
Ready for the next challenge?
Absolutely. We've covered a lot of ground, from those tricky clinical presentations and lab diagnostics to the ever-evolving world of antibiotic resistance. Now it's time to explore the front lines: prevention, control, and treatment strategies.
I'm ready to arm ourselves with the latest knowledge. Let's dive in.
All right, microbathers, we're back. We've delved into Staphylococcus aureus' tricks, its weaponry, even its ability to outmaneuver our antibiotics. Now it's time to turn the tables. Let's talk strategy, prevention, control, and how we fight back when those infections take hold.
Absolutely. We've seen firsthand how persistent and adaptable S. aureus can be, so our strategies need to be just as dynamic.
Agreed. Let's start with prevention, especially in those high-risk healthcare settings where S. aureus seems to thrive. What are our best defenses?
Well, it may sound simple, but hand hygiene remains our absolute bedrock.
The golden rule of infection control.
Exactly. Frequent and thorough hand washing, especially between patient contacts. It's a simple action, but it can truly break the chain of transmission.
It's amazing how often the basics make the biggest impact. But what about those cases where we're dealing with known S. aureus carriers? How do we minimize their risk of spreading the bacteria?
That's where decolonization comes in. In certain high-risk scenarios, like patients about to undergo surgery or those with indwelling medical devices, we might try to eliminate S. aureus from their nose and skin. Topical mupirocin is often used for this purpose.
So we're essentially trying to evict those unwanted bacterial tenants from their prime real estate.
Precisely. But of course, even with the best prevention efforts, we know infections can still happen. So let's talk treatment. What are our weapons of choice when S. aureus breaks through those defenses?
Bring on the antibiotic arsenal.
Well, before we deploy those, we first need to know what we're dealing with. Is it a methicillin-susceptible S. aureus, MSSA, or the more formidable methicillin-resistant strain, MRSA? That distinction guides our treatment strategy.
Okay, so for MSSA, we can still rely on those trusty beta-lactam antibiotics, right?
Right.
I'm thinking of drugs like flucloxacillin.
Flucloxacillin is still a solid choice for MSSA as long as the strain is susceptible. We also have other beta-lactams in our arsenal, like cephalosporins and carbapenems, but we always have to be mindful of those emerging resistance patterns.
Resistance is a constant worry. And for those MRSA cases where our beta-lactam wagons are essentially useless, what are our options? I remember learning about vancomycin as the go-to drug for MRSA, but I know there are concerns about emerging resistance there too.
You're right. Vancomycin has been our mainstay for MRSA for a long time. But those increasing resistance rates are definitely a concern. When vancomycin fails, we have to turn to alternative antibiotics, like daptomycin, linezolid, or ceftaroline.
So it's a constant battle, adapting our strategies, searching for new weapons in this microbial arms race.
Yeah, it is.
But antibiotics aren't our only tools in this fight, right?
Right.
What about other aspects of managing S. aureus infections?
Absolutely. Supportive care is essential, especially for those severe cases. Intravenous fluids, pain management, wound care, those can be critical. And sometimes surgical intervention might be needed to drain an abscess or remove infected tissue. It's all about a holistic approach, considering the whole patient picture.
It takes a team, doesn't it? Microbiologists, clinicians, surgeons, all working together.
Teamwork is essential, but ultimately our best weapon against S. aureus is prevention.
I completely agree. Prevention's always better than a cure, especially with such a cunning adversary.
Exactly. Staying vigilant with infection-controlled practices, educating patients and healthcare workers, and continuously researching new prevention strategies, that's how we stay ahead of the curve.
Well said. I feel like I've leveled up my S. aureus knowledge today. But before we wrap up, I have a few exam questions for our listeners out there.
Excellent idea. Always good to put that knowledge to the test.
All right, here we go. Question number 1. What are the key characteristics of small colony variants, or SCVs, of S. aureus, and why are they clinically significant?
Great one. SCVs are those sneaky, slow-growing forms of S. aureus. They often lack the typical golden pigment and can really make things tricky. They're significant because they can linger in the body, dodge antibiotics, and cause those difficult-to-treat chronic or recurrent infections.
They're masters of disguise. Okay, question number 2. Name 3 important adhesins produced by S. aureus and describe their role in infection.
Let's see, we could choose from quite a few, but I'd say those fibronectin-binding proteins, or FMBPs, are key for grabbing onto host tissues. Clumping factors, especially CLFA, are experts at binding fibrinogen and promoting those troublesome clots. And then there's protein A, which actually binds to our antibodies in a way that helps the bacteria evade our immune system.
Sneaky, sneaky. All right, question number 3. Explain the mechanism of action of pantin-valentine leukocidin, or PVL, and its association with community-acquired MRSA.
PVL is like a tiny but powerful weapon. It's a toxin that essentially punches holes in white blood cells, causing them to burst. It's often found in those aggressive CA-MRSA strains, contributing to severe skin infections, necrotizing pneumonia, even sepsis. Definitely a toxin we don't want to encounter.
Okay, 2 more to go. Question number 4. What are the primary strategies for preventing and controlling S. aureus infections in healthcare settings?
Here's where those infection control fundamentals come in. Hand hygiene, always at the top of the list. Careful antibiotic stewardship to prevent resistance from running rampant. Isolation precautions for infected patients. And meticulous environmental cleaning and disinfection to eliminate any lurking S. aureus cells. It's all about staying vigilant and working together as a team.
Teamwork makes the dream work. And finally, question number 5. What is the role of biofilm formation in S. aureus infections, and why does it pose a challenge for treatment?
Biofilms. Think of them as S. aureus's protective fortresses. A slimy, cohesive layer that shields the bacteria from both our antibiotics and our immune system. It makes biofilm-associated infections incredibly difficult to treat, particularly those involving medical devices. It's one of the toughest challenges we face in the fight against S. aureus.
A formidable defense indeed. Well, there you have it, folks. 5 challenging questions to test your S. aureus expertise. We've covered a lot in this deep dive, from its basic biology and crafty virulence factors to the latest on diagnosis, treatment, and those critical prevention strategies. It's been a wild ride through the world of this remarkable microbe.
It has. Staphylococcus aureus is a truly fascinating and formidable opponent, constantly pushing us to learn and adapt.
But armed with knowledge, vigilance, and a healthy respect for this remarkable microbe, we can continue to fight back and protect our patients from its wrath. Thanks for joining us on this deep dive into the world of Staphylococcus aureus. Until next time, keep exploring those microbial mysteries and stay curious.
See you next time.